My name is Mongoy Buguero, and as part of my PhD, I create computational models of immune signaling and cell growth in uterine disorders. Here I'll talk about my work creating a mechanistic computational model of IL-8 signaling in neutrophils. Through creating this model, we were able to quantify the rates of processes that affect IL-8, and we have used this model to explore the role of these processes in neutrophil activation. So we're interested in IL-8 specifically because it is important for healthy menses. But IL-8 is also involved in the development of gynecological cancers and even non-cancerous uterine disorders like endometriosis. Although we think of IL-8 typically as being produced mostly by monocytes to recruit and activate neutrophils, studies have actually shown that neutrophils can also secrete low levels of IL-8. However, one thing is still unclear. Can this neutrophil secretion of IL-8 affect the activation of these same neutrophils? To answer this question, we model IL-8 signaling on neutrophils using ordinary differential equations. These equations predict how the levels of IL-8 and of their receptors on neutrophils would change in response to ongoing biophysical processes. Some of these processes include neutrophil secretion of IL-8, ligand receptor binding, and receptor trafficking. We estimated the rates of these processes through optimization using our model and experiments from a published study. Now, earlier, I had mentioned that the importance of neutrophil secretion was still unclear. One interesting result from our modeling was that although the rate of neutrophil secretion was low, it was not negligible. Without including neutrophil secretion of IL-8, the model was unable to fully capture the dynamics of receptor resurfacing or the increases in IL-8 levels that have been observed in experiments. In my poster, I describe our approach to optimization, and I explain how we use sensitivity analysis and additional model simulations to characterize how these biophysical processes can work together to impact IL-8 signaling. These findings can help us develop computational models to further understand the role of IL-8 signaling in both healthy and diseased uterine tissues.